Hi, everyone. I'm Anna, application engineer from Joe, Singapore. Today, I'm presenting this topic, automated array tomography in 3D SEM for biological applications. So this is the outline for today's presentations, which cover the background of the 3D SEM. Three techniques will be reviewed here. First, serial block phase. Second, focus ion beam SEM. And third, automated tape collecting ultramicrotomes. Secondly, I will introduce the automated 3D array tomographies, and finally present the solutions from 3D acquisitions to reconstructions. So what is the motivations behind 3D SEM? In the optical microscope, using the photon source that it can produce large volume for at few hundred microns up to several millimeter depth. And with this advanced confocal laser microscope, it can give much more better resolutions, and this has entered into the electron microscopy range, whereby we can observe synapse in few hundred nanometers. However, if we look at this example of the confocal images here, mainly the image are frozen stack, and there are no detailed structure information provided. So SEM and TM open up the view of three dimensionals into ultrastructural analysis. 3D TM can offer the highest resolutions for the tissue volumes. It can allow to observe the smaller cellular structures at sub-nanometer resolutions. For example, here in these 3D tomographies, we can observe the cross-sections of the cilia, showing the 9+2 microtubules very clearly. However, there is a limit into the volumes, whereby it depends on the number of sections that can be imaged and the section thickness that allow the electron to pass through. The field of view is seriously restricted. It is difficult to observe the whole image of the cells or tissues. So 3D SEM can fill in this gap between the confocal and the TM by allowing to produce a large 3D SEM volumes at few hundred microns and sufficient to observe the small organelles at nanometer resolutions. Therefore, 3D SEM can offer high resolution with large volume and wide fields of view. Before I go into the details, let me explain the basics of 3D SEM. So we have thin sections prepared from the ultramicrotomes, and we image these sections under the SEM using the backscatter detectors. The backscatter image are then reverse contrast to result in the same as the TM image, whereby the feature is showing into the dark and the white contrast is the empty space. The continuous sections are imaged to form a series of 2D stacked images. By using a software, subsequent applications of image processing, alignment, stacking, and modeling step help to generate these 3D models. The volume is defined by 3 dimensions, X, Y, and Z, while the XY resolution is the smallest distance between two objects in X, Y respectively. The Z resolution is the section thickness, which depends on the smallest section thickness that we can produce. For dominant features into the tissues, we can actually perform segmentations with multi-colors in order to highlight the features to be more outstanding and for easy to interpret. For example, here I have a neuron cells segmented out from the brain tissues displayed in the red structures. So from here, we can observe the cell body and the dendrites. We can see how the dendrite is branched out from the cells to all the direction into the tissues. Also, for analysis, we can perform to measure the length and the dimensions of the dendrites. For complex biological structures, we can perform multi-segmentations. For example, here I have 4 neurons displayed in 4 different colors in order to map the complicated neuron circuit into the tissues. In this case, we can study how the cell is communicating to the brain. Next, I will review the 3D SEM technique, each with their advantage and disadvantage. To overcome the drawbacks of the manual serial block phase TM, this serial block phase SEM is introduced. A miniature ultramicrotome is installed into the SEM chamber, whereby it can perform to slide the sample and SEM observations. So we have a tissue block attached to the stage, and the block surface is cut by this diamond knife. The new fresh area of the block surface then is imaged by the backscatter detectors. So this process is automatic, and the diamond knife cut deep into the tissues. The serial block phase SEM can produce a large dimensions, especially in the Z dimensions at few hundred microns. This is much better than the TEM volumes in producer tomographies. The resolution in XY depends on the SEM resolutions or SEM performance, while the Z resolution is the section thickness, and this can be varied from 25 to 80 nanometers. This depends on the cutting properties we set in during the cuttings. The disadvantage from this method is it is a destructive method whereby all the section is lost after the imaging. In addition, we need to apply additional heavy metal staining to the block in order to improve the contrast and the conductivities. Focused ion beam SEM bridge between the serial block phase and the TM tomography by produce a slightly larger Z dimensions at 20 microns. This is slightly better than the TM volumes. However, it can offer the highest resolutions at 10 nanometers if compared to other 3D SEM techniques. In the chamber, there are two emission sources. One is on the top of the column, producing the electron to image the block surface, while the other on the side of the column, the gallium ion beam to mill the block surface. This fine focused ion beam can mill as thin as 10 nanometers. The focused ion beam SEM can cut any materials, even hard materials that is impossible by the diamond knife. For beam sensitive samples, there is an option of cryo FIB at low temperature is available. Same as the serial block phase, this technique is a destructive whereby all the section is lost after the imagings. In addition, with the small ion beam here, it actually mill a very small areas. The field of view is around 20 microns, and this area is increasing with the milling times, means that if we mill for a larger area, it will take longer time. Eventually, it will cause another issue, charging accumulate onto the block surface. In order to produce large volume, there is this automatic tape collecting ultramicrotome introduced. The automatic tape collecting device is attached to the ultramicrotomes. The tissue block is cut by the diamond knife, and the sections that produce are collected on this long, movable Kapton tape. The sample on the Kapton tape then are cut into a small stripe and placed on this silicon wafer for SEM observations. This method can result in a big areas up to 3 millimeters, and this value can be bigger if we use a bigger diamond knife. The Z is depending on the effort. The effort preparations is intensive laborious, and with this complicated setup, the user need to be skillful and experienced in the ultramicrotome to produce to perform perfect alignments and to produce the ribbon in a straight line. The advantage from this technique is the section is preserved after the imagings. That means we can keep the samples. Today, I'm introduced this automatic 3D array tomography. Ribbon of serial sections are collected in order arrays and subsequently imaged on SEM by automations. This technique can produce large field of view for bigger areas and large volume for more data at high resolutions. The process is fully automated from serial sectioning to 3D reconstructions. This automatic process allows more easy, faster, and reliable way to acquire tissue volume for ultrastructural analysis. If compared to the FIB and the serial block phase, the sample is prepared and imaged simultaneously. But in this method, array tomography here, the section is prepared and we have the options to stain the sample or perform immunolabelings, then image later onto the SEM. Therefore, the sample can be stored or preserved for a period of time, and we can always re-image for different interest area and for different magnifications. The array tomography also enables for corrective light electron microscopy work, whereby we have options to do multiple fluorescent labeling, and finally corrective to the 3D SEM. So this slide shows the full workflow from the sample preparations to the imaging and analysis integrated by Leica and JOES. So the sample preparations part is done onto the Leica products, while the imaging and the analysis is performed on JOES SEM and array tomography software. So first we have a tissue block, and we trim to a smaller area close to our region interest and into the shape and size that can be fit into the ultramicrotomes. This process can be done by these auto trimming machines here. With the consistent trimming step, it can produce a very smooth surface smooth edge, and the shape that perfectly parallel to the knife edge. The trimming block then is transferred to the ultramicrotomes. In this case, we use the ATOS from Leica that dedicated for 3D ultramicrotome. These machines can perform automatic serial sectioning and pick up with minimum human interference. Long ribbons in order arrays and with uniform thickness are produced, and eventually automatically collected onto the silicon substrate. The sample then is transferred to our Joe FESEM. This FESEM can produce a high quality backscatter image in terms of contrast and resolutions. Finally, by using the software, we can perform auto reconstruct from 2D data into 3D models. The whole process is fully automated, whereby it allows few thousand of sections can be easily prepared with minimum skill of experience onto the ultramicrotome and onto the SEM operations. Give me this opportunity to update our Joe latest SEM portfolio. We can provide a range of products from a small mini compact benchtop tungsten SEM to ultra high resolution field emission SEM. JSM IT800 Super Hybrid Lens is our latest FESM models. It gives the highest resolutions at 0.5 nanometer at 15 kb. All the SEM model here can perform both imaging and analysis, each with their unique features and dedicated applications. In addition, we can perform 3D surface analysis using the Smart View map. Also, we provide a 3D tomography solutions. 4 software work together to reconstruct for the 3D SEM. This slide, I will highlight some of the features of our high-end FESEM. The electron gun is an inline shortcut gun, and with this conical shape of the condenser lens, the gun tip is placed close to the electrode, close to the magnetic fields. This allows the electron come up from the gun to be efficiently focused to the SEM columns. This results a large current, maximum at 500 nanoampere. This is 10 times larger than the conventional SEM. It results in the greatest brightness and intensity. The electron gun has a long-term stability with less than 1% fluctuations. This is good if we are running for a long process time analysis. Also, because all the electrons are fully utilized here, the electron gun has a long lifetime with 3 years warranty. In practically, our electron gun can be used for longer, even up more than 5 years. There is this new electro-optic control system into the SEM that can provide highest level of automations like autofocus, auto stigma, and auto beam alignments. This makes the SEM to be more easy to operate, especially for beginners. The high accuracies of automations also benefit to the 3D auto acquisitions. Joe patented this aperture angle control lens here, whereby it can improve the resolutions at high probe currents. Usually when we increase the probe currents, the beam size is bigger and eventually the resolution becomes poor. But with this aperture angle control lens here, it can help to maintain the beam to be small and focused, even we change from low probe current to high probe currents. On the other hand, our objective lens is a super hybrid lens, combinations of electrostatic field and the electromagnetic field. This result excellent resolutions at low accelerating voltage. There are various of high efficiency and high sensitivity detectors available here for topography, morphology, and composition imagings. For 3D SEM, backscatter detectors are mainly used to image the sections. Now I will demonstrate how our system can perform auto acquisitions to 3D reconstructions. Okay. So this is a workflow for the automatic image acquisitions. This table shows the step-by-step. First, we have the optical image showing all the samples on the wafer. In the second step, we perform SEM mapping on all the ribbons on the substrate. This is an example showing 2 ribbons on the silicon wafer. In the step 3, we acquire a low magnification image that allows us to identify each of the sections. This image is taken at a quick scan and low resolutions. The images are mainly used for orientations and to locate the region of interest for the 3D reconstructions. In the final step, we acquire a more high magnifications of our ultrastructural at same orientations. This image is taken at a slower scan and high resolutions. So we have a sections we prepare from the ultramicrotomes and place on the silicon wafer. Before we load the samples into the SEM chambers, we capture the image of whole sample using the optical microscope or the phone cameras, showing all the ribbon sections on the substrate. Then on the SEM side, we open the tomography recorder software and create the projects file here. Then in the step 1 here, we import the optical image that we have acquired before and loaded into the software. This is the imaging condition in step 1. The accelerating voltage is 5 kV, working distance 10 millimeters, and in step 1 we use a LED detector for the imagings. Next, we add the observation area all over the ribbons as shown in the yellow box here. Then we can perform to start the SEM imaging for step 1. In the step 2 here, the optical image is automatically replaced with the SEM image. So here showing is the SEM image of all the ribbons. And in the step 2, we change the imaging conditions. The working distance is made to shorter to 4 millimeters. And in step 2, we use a backscatter detector to image the individual sections. In this step as well, we need to design the array samples to register the observations area. So this is shown in the next slide, how we can register the observation point and identify the number of sections for 3D reconstructions. So the plus marking here is the current SEM stage positions, which is our observations point. So we need to do a labeling PQ on 2 edges of the sections. And this is shown in this video here. So for the first sections here, we label PQ and register the first observations point manually. And for the following sections, we just need to label PQ and the observations point will be automatically registered. After a few labelings, then we can push on this play button here and software will automatically register for the rest of the sections. So here we can identify there are total numbers of 30 sections registered for the first ribbons. After we design the array samples, then we can proceed to start the SEM imagings. In the last step, step 3, we open one of the image that we acquired in the step 2. And show here, we can add the observation area onto the ultrastructural that we are interested for reconstructions. Okay. Before the imagings, the software will perform to correct the image shift and rotations for all the sections. On the top here, it shows is before the corrections. We can see the double image, and this is after the corrections. Finally, software will automatically acquire a series of 2D SEM image of our ultrastructural at high resolutions. After completion of the acquisitions, we can add another observation area on the same sections from step 3 without going back to step 1 or step 2. Therefore, we can perform a multiple 3D SEM data on the same sections for our study. The 2D data that we acquire from the SEM then are transferred to an offline PC, open with the Stacker Neo software to reconstruct the 2D model data into 3D models. This video will show how the Stacker Neo can perform to transform the 2D data into 3Ds. So first we open the projects and we upload all the SEM images into the software. And now we check one by one the images and we can choose to unselect any imperfect or damaged sections from the reconstructions. Also, we can see the continuous image by using the slideshow functions. After that, we need to input the section thickness, which is the Z resolutions. Now we can perform the alignments. We draw a small rectangle and place it close to the region of interest. And we press Start Alignments. Software will automatically correct the shift and rotations for all the images. Now we check the image after the alignments. If the alignment is good, then we can proceed to stack all the images. To do the stacking, same thing, we draw a small rectangle and put onto the region of interest that we want to reconstruct. Then we press on the Start Stacking. software will automatically stack all the 2D images into 3D volumes. Okay, so this is the results we have, and we can display the result in XY, XZ, and YZ plane. In addition, we can also observe the cross-section image from the volume data. Also, there is this visualizer button here that can transfer the 3D volumes into another software visualizer able to present as a 3D models. So this outline shows the step-by-step of the 3D reconstructions. For these 3D models, we can also perform to highlight the ultrastructural into different color by using the segmentations. So next video will show what we can do with our 3D model using the visualizer able software. So same thing, we open the 3D volume that we created from the stacker software and upload into this visualizer able software. And we can choose to configure the brightness. And here we recommend to use a binning one, especially if we have a big data. Okay, on the software, we can see the 3D models, and we can rotate the 3D model by using the mouse. We can move out to see the overview and move in to see to zoom in to see the clear features. And we can move the features and to look into the 3 dimensionals. In addition, we can also perform cropping in 3 directions, X, Y, and Z, to see the internal structure or cross-sections of our features in our 3D models. Now the 3D models is showing into the grayscale, and we can proceed to apply color. There are a lot of selections of color here we can choose. Okay, and we can also proceed to adjust the volume color and the transparency. We can also transform the 3D models into the 2D plans by using this function slice. Okay, now the 3D models is changed to a 2D plan, and we can perform to observe the slice by slice in X, Y, Z directions. This allows us to gain the further insight of our structure into the 3D volumes. On the other hand, there's another features here, ISO surface, that allow us to create the surface as a mesh in order to study the topography of our samples. So now I have examples of a 3D reconstructions of the nervous system. And here we can observe the myelin, and the myelin is surrounding the axon in 3 dimensionals. And this is a cross-section view. And also we display in a 2D plan to see the structure inside the myelin in cross-section view. Besides that, we also observe the morphology change of the myelin throughout the space. And here is another video showing a 3D models in color. We segmented up the axon out from the myelins. Okay. Then we combine back the axon to the myelins for the whole 3D volumes. And this is performed by auto segmentations, whereby the software can automatically separate the dark contrast from the white contrast throughout the volumes. The dark contrast is the myelins, while the white contrast is the axons. And also we can select to display only the myelin in order to study the myelin structure in the 3D volumes. Here we can observe the morphology change of the myelins in the volumes. I'll end the presentations with this last video here. Okay, hope you all will enjoy. Although SEM cannot achieve the resolution same as a TEM, but with the modern FEA-SEM technologies at nanometer scale resolutions, actually it is sufficient to answer many of the biological questions and applications. I come to the end of my presentations, and I thank you for your attention.